Hello friends, welcome to Expert Guidance. Today in this video we'll be covering the topic of biological molecules of the Unit 2 Structure and Function in Living Organism. Now in this video we'll be covering the important biological molecules which are the main components of the food, the carbohydrates, proteins and lipids. We'll be looking over the structure of these along with the differences. We'll also look over how you can distinguish and test on the food for the presence of glucose, starch, protein and fats. And then we'll come to the topic of enzymes. We'll look over how the enzymes function, the properties of enzyme, and how the activity of the enzyme be altered by change in temperature and pH. Now this is a very important unit because there are a lot of graphical questions on enzymes that are asked in the exam, and the concept of enzyme is again very important. So I would recommend you to watch the video till the end so that we'll make sure that you are not leaving any of the important bits of this unit. So let's begin. Now, let's see what are the components that we take in with the food. We take carbohydrates, proteins and fats. Carbohydrates are present in bread, pasta, cereals and rice. Proteins are present in pulses, chicken, meat, poultry, egg, beans and nuts. And fat is present in butter, oil, margarine. Carbohydrates are composed of sugars. Proteins are made up of amino acids. And fats are made up of fatty acids and cholesterol. And this is what these components get digested into. The enzyme for carbohydrates are carbohydrates. Example, amylase. For protein it is proteases, and for fat it is lipases. Carbohydrates are digested in mouth and small intestine. Proteins are digested in stomach and small intestine, and fats are digested just in small intestine. Carbohydrates are the principal source of energy. They are fuel for respiration. They can be storage molecules like starch and glycogen, or structural molecules like cellulose in plants. Proteins are the components of muscles required for growth and repair. They're the components of enzymes and they are also components of some of the hormones like insulin. Fats are used to insulate the body. They are the reserve source of energy, and they also makes the cell membrane. Okay, so you should know what are the different components of food, their sources, where they're digested, and what is their function in the body. Okay, so now since we are doing digestion, and digestion is chemical, that is, it involves the use of enzyme, you should know what are enzymes. So make sure you remember this definition and this lock and key model, very, very important. So I'll just take you through it. So enzymes are biological catalysts. So I think in chemistry, you remember what are catalysts? Catalysts are those substances that increases the rate of a reaction without being used up. So if there's a catalyst which are used in biological reaction, we call them enzymes. So enzymes are biological catalysts that increases the rate of a biological reaction without being used up. Now, what do enzymes do that increases the rate of reaction? They actually provide an alternative route to a reaction which works by lowering the activation energy. And enzymes are chemically proteins, so they are very sensitive to heat, pH and temperature. Okay, now this is the structure of the enzyme, and the enzyme has a specific shape, and it has a site called the active site. Active site is a site where substrate molecules bind to it. Now substrate and the active sites, they have complementary shape. Remember to use the word complementary. Most of the time in the exam, students say substrate and active site have same shape. No, that's wrong. It's like a lock and key. The substrate and the active site, they have a complementary shape. The substrate binds to the active site, forms an enzyme substrate complex. The enzyme substrate complex react to form an enzyme and the product, and it disintegrate to give the product back, and your enzyme gets regenerated. So remember, enzymes are highly specific due to the active shape. As the active shape has a specific shape which is complementary to the substrate, so the specific substrate molecule can fit into the active site of the enzyme. Now, what are the factors that affects the enzyme activity? Temperature and pH are one. We'll be discussing that. But the substrate concentration and enzyme concentration also affects the rate of reaction. So as we increase the substrate concentration, there'll be more molecules to bind with the active site. Therefore, the rate will increase at the start. But after that, it levels off because all the enzyme active sites will be used up, and then enzymes will become a limiting factor. The same thing work with the enzyme concentration. As we increase the enzyme concentration, there'll be more active sites for the substrate molecules to bind to. But after that, further increasing the enzyme concentration will have no effect because then all the substrate molecules will be used up. So substrate will become limiting. So the graph will level off. Now for the temperature, as we increase the temperature, the rate of enzyme catalyzed reaction increases because the particles gain kinetic energy. As per chemistry, they'll collide more the collision, more the chances of reaction. It increases up to a point, and this point is called the optimum. Optimum is the point where the enzyme activity is the highest. But after that, the rate will drop suddenly. Why is that? This is because the enzyme gets denatured. Now, what is denaturation? Now, denaturation is when you heat any specific enzyme due to being protein in nature, their bonds break. So they change the shape. Now, when they change the shape of the active site. So the substrate molecules can no longer bind to it, decreasing the rate of reaction. So denaturation is change in shape of the enzyme with a change in pH or the temperature. The same effect happens with the pH. As you increase the pH, it increases the rate of reaction up to the optimum, and after that it drops. It doesn't drop that suddenly like temperature, so we make like a bell-shaped curve. And then it again drops. Why? There's a denaturation because the shape of the enzyme changes. Now for the temperature, the optimum is around 37 to 40 degrees because that is the normal body temperature. But for the pH, different enzyme has different optimums. Like stomach enzymes, they work best in acidic condition, and the intestinal enzymes, they work in the alkaline condition. Okay, so I hope this graph is clear to you. You should be able to draw this graph in the reaction in the exam, or if this graph is given, you should be able to explain all this trend, why there's an increase, what is an optimum, and why there's a drop. And I hope you will remember it. Okay, so what is the next step? Next step is check the specification. Make sure whatever thing is there in the specification is crystal clear to you, and do exam questions on this topic. Okay, so I hope you have understood this video well. If you still have any doubts, leave a comment below. We'll reply you as soon as possible. Or you can go to our website where we have 24/7 chat support, where you can post any queries before your exam and you can get instant reply. You can go to my website where you can find the notes of this as well. The link is mentioned in the description box below. If you like this video, then choose subscribe to our channel, and do not forget to like, comment, and share this video. And if there's any specific topic you want me to make a video on, do leave a comment below and I'll make sure I'll put that up before your exam. Okay, so please go over this topic very, very seriously. It's a very important topic, and I'll see you next in your next video. Till then, happy revising.